All right, we're going to take a quick look at replication. This is more for the core in the SL level, but just the basics of replication depend on the fact that DNA structure involves these complementary base pairs, right? So if we have an original parent molecule of DNA, right, which is made up of two strands, because we know DNA is a double helix made of two strands. And in that parent molecule, the strands aren't identical to each other. They're actually complementary to each other, meaning the A's and the T's go together, the C's and the G's go together. So if you were to pull apart these two strands, they're actually not the same. They're complementary. However, that complementary nature still allows for identical replication, because if you come in and then add the complementary base pair to this strand and the complementary base pairs to this strand, as you can see, this is happening here, so the A's and the T's and the C's and the G's and so on, then in the end, you end up with two DNA molecules where one strand is an original parent strand, and then the other one is the complementary strand to that. We call this semi-conservative replication, and we're going to look at some interesting experiments that confirmed semi-conservative replication in another screencast. Okay, so first of all, this is a lot more information on this than you have to really totally absorb, but I just want to point some things out. First of all, when there's a whole bunch of enzymes involved in the process. And for those DNA strands, when they pull apart, when the two strands within the DNA molecule pull apart, they will pull apart and create this bubble or a replication bubble. So you might hear that terminology of a replication bubble. And at either side of the replication bubble are what we call replication forks. And that's where all the enzymes are going to be working to add the complementary base pairs. And it happens in both directions. So we're going to get replication happening this way and that way. In eukaryotic cells, there's going to be multiple bubbles because there's so much DNA that has to be copied. In prokaryotic cells, often it's just a single replication bubble. Okay, so now this happens actually really, really quickly, 50 nucleotides per second in human DNA. It's pretty amazing. And it does turn out, and I'm going to point something out to you that you don't really have to pay too much attention to at the SL level, but it's going to be very important at the HL level, is that I said that it moved in both directions on the replication bubble, but it's interesting because it can only build easily in one direction because the DNA strands are directional because of where the phosphate lies in the nucleotide. We call that the 5' end. And you can only add to the end that doesn't have the phosphate. And so you'll see there's going to be two different ways. There's going to be a leading strand and a lagging strand that's being made. But for the core, for SL, you only need to really pay attention to the details of creating the leading strand. All right, so let's take a look at those details. So here is part of this would be the whole replication bubble here, but here's the replication fork. Okay, now, as I said, there's a whole bunch of enzymes involved. The first one that you would want to mention is helicase. Helicase, what that does is it's going to go in and disrupt the hydrogen bonds that exist between the nucleotides. Because, let's say you have an adenine here and a thymine on this side. Those are actually held together with hydrogen bonds, if you remember how to draw your DNA molecule. And so these two strands are held together by hydrogen bonds, but the helicase can disrupt those and open up the replication fork, creating that bubble. So that's the job of helicase, is to unzip and essentially unwind unzip the DNA molecule. Now, another important aspect here, there's these things called single-strand binding proteins, and these guys come and clamp down so it can't shut back again, because once this helicase goes by, we don't want the hydrogen bonds to come back and attach back here again, so it keeps it open. And then there's also a molecule called DNA gyrase, and that actually takes some pressure off because it can end up supercoiling if you're like pulling the two strands apart in front of the helicase. And so what that'll do is it'll open up and reattach things quickly to be able to keep that tension going. If you have two strands wound together and you give it a try and you pull them open, it actually creates a tighter winding on either side of that bubble. So the DNA gyrase keeps that pressure. So it relieves the strain of that unwinding by breaking and then reforming some of those bonds. All right, so helicase is the first to unzip it. The single-stranded binding proteins keep it open, and the gyrase keeps it from, supercoiling on either side. Now, to start with the concept of complementary base pairing, always make sure you bring up that exact term when you're talking about it. We need to involve first this enzyme called primase. And what primase is going to do, primase is like priming the pump. It's going to get the replication process started. And it's going to add a short RNA primer to the open strand. So it's going to come in and add, through complementary base pairing, some RNA. And so if this is T, this would be A. If this was, G, this would be C, and so on and so forth. So it's going to add the complementary base pairs, but as RNA right here, just in a short little bit. Once it creates the primer, that primase can fall off. Now, once that primer's there, then the molecule that's responsible for actually adding the DNA nucleotides along the way, that can start its job, and that is called DNA polymerase. So DNA polymerase is going to link the nucleotides together to form a new strand, and so it's going to follow behind the helicase as it opens it up and just add, through complementary base pairing, the appropriate nucleotides one after the other, and just follow through. And so you can see it's just getting longer and longer and longer as it follows down behind the helicase. We're not going to get into it in this one, but this strand on the other side has to be built in shorter fragments because it can only be done in one direction. But this part of the leading strand is the most important part you need to be able to talk about.